So guys, today we will learn that how we can extract the Go terms of specific number of genes or specific type of genes. Why we use the Go terms? Actually, Go terms come from the uh, gene ontology. That is, that if we want to know the function of a gene, because genes are known for their functions. So we want to know, see that what sort of functions are associated with gene. For that, we can make use of the ensemble genome browser. Actually, Biomart is a very important tool of ensemble genome browser, by using which you can know the Go terms of your genes. So let's we go to the ensemble Biomart. Ensemble Biomart. We wrote it here in the Google. So here comes the Biomart. So you can see that Biomart is somehow it's not functioning. There is some problem with the website. It's down. So you can use the mirror sites. The page took us to the mirror site. Let's we use the uh, uh, US East Ensemble org site and see if it's functioning. So you can see here is the Ensemble Genome Browser, one of the most widely used genome browser. So here is the Biomart link. We can go to the Biomart. So here we have this Biomart page. So we select here the ensemble genes because we are going to deal with the genes, and we will use the human genes because we are at the moment looking for the Go terms of the human genes. You can select whichever species you are looking, which is your really species on which you are working. You can look at the species of your interest from this top-down list. Now we select here the filters. In the filters, you can uh, click on the gene and you can input whatever number of genes you want to. You will put here the ensemble gene IDs of the genes of which, or the genes whose Go terms you want to know. But at the moment we want to, actually we are looking for the Go terms of the whole genome of human, that is all the genes of the human, so all the protein coding genes of human. So we go to the gene type and we will go to the protein coding. These are protein coding genes, fine. So we get it, and now we will go to the app. Once we have selected our filter, that is the gene type, that is protein coding genes, we'll go to the attributes, and in the attributes we will go to the external. First, before external, we go to the gene. We only want gene stable ID, so we unselect the other irrelevant things, and we want the gene name also. Let's say this is gene name. So these two attributes we have selected until now, and they can be seen here: gene stable ID and gene name. And then we will go to the Go term, which comes in the external section, which is titled as the external, and here we have the Go term. So we will we will select here the Go term name and the Go term domain. So the Go term name will tell you about the function of the genes, and Go term domain, actually there are three Go term domains which are usually which the function of the gene has been categorized into three domains. One is the location of the genes, second is the process, a general process what the gene is associated with, and the molecular function which gene is performing. Fine. So we have there four attributes: gene stable ID, gene name, a Go term name, and Go domain. We go to the results then. This is the results. And in the results section, it's better always, because we are although it is already showing us the results, but it's always better if you are going to deal with a very large data set. You go to CSV file, and because CSV files are easy to handle for dealing with the large data sets, and then we uh, and it will start downloading the CSV file. You can see that it is downloading it. It will take some time because it would be quite heavy file, because we are here extracting the gene um, terms or Go terms of all the genes of the human genome, all the protein coding genes of the human genome. So we can go to our download material. We can open the download material by using Notepad Plus. You should also download and install the Notepad Plus onto your PC or your laptop, because it is the type of notepad which is specifically used for dealing with the large data sets. So I open this, and actually I have, so if I file, I open, and my this is my download file in the downloads. I open the whole data of Biomart, which is downloaded in the download folder in the form of text file or notepad file. I open it, and it will now open. So this is now the whole file is opened here. Now this file, you can see it is not in the a simple column form, which is more easier to understand. So it will be good that if we copy this data and put it into Excel file. So actually this is a huge data. This is actually, I would say, around 20 lakh genes data, or 20 lakh entries for the Go terms. It's not possible to put this whole data into the Excel. What we can do, we can uh, copy it, and we copy it piece by piece, and then paste it over there in the Excel. So what we can do, we can copy at least 4 lakh genes each time. So in five, by repeating this process by almost 4 to 5 times, the whole data will be then pasted onto the Excel. So this is, we can go to, so this is around 4 lakh, no, this is 42,338. Let's we go to 1 lakh, 19 lakh, 2 lakh, 2 lakh, 2 lakh, 3 lakhs. This is 4 lakh. So until 4 lakh 9,000. This is around. We copy this part over here. Go to the Excel. We put it into the Excel here. Fine. So this is 4 lakh, and then the remaining you can do it again. For more lakh, that is around 8 lakh in total. We put it over here. Control V. Fine. So keep in mind that up till now, up till this date, in Excel you cannot put more than 10 lakh entries into one single file, or one single page, I mean, or one single sheet rather, of the Excel. So you can, for the other um 9 or 10 lakh of the genes, because there are around 20 lakh genes or 20 lakh entries, you can generate another sheet, open another sheet, and then put the remaining over there. So we select 4 more lakh, which will be until 12 lakh. Okay, we can do it over here from this is 12 lakh. So it's very easy. You should, while you are dealing with the Excel, it's always good that you know certain keys also for selecting the data. So if we uh, want to select a big chunk of data, it's always good to use the shift key. So I select, for instance, I selected the gene starting from the 8 lakh, and when I reach to the 12 lakh, I simply put, I click the shift key and then make a left click, and you see the whole 4 lakh genes are selected from the previous click till this click. That is from the 8 lakhs to 12 lakh gene entries. We'll put it over here and we select it over here. Fine. And in this way, you can copy paste all of your notepad data into Excel. Now once you have done it, what would you want to change it into tabular form? It's very easy. You simply select one of the column, I mean this column, and you go to the data and you you choose the text to column tab, you choose the delimited tab, go to the next, and choose, because this is comma separated data, that's why this is known as CSV file. We use the comma section with comma tab is selected, and we use the next, and we put the finish. And in this way you will see the whole data will be changed into column form or tabular form, in which all the separate entities will be put into the different columns. So you can see this is gene stable ID, this is gene name, this is the Go term name, this is the Go definition, and this is the Go domain. Now if you want to select any specific specific function, or you want to select those genes which are involved in specific function, you can simply use the filter option from here, and you can select the genes of your interest. Fine. Good. Now hopefully this will help you to, this whole exercise will help you to extract the gene ontologies of your genes. In that way it will be easier for you to know what your gene of interest, what function your gene of interest are performing. Fine. Thank you very much.